Hello, this is Dr. Stephen Pipe, Professor of Pediatrics and Pathology at the University of Michigan. Well, this is in regards to a presentation that we just heard on a first-in-human gene therapy study of a unique AAVhu37 capsid vector technology applied to severe hemophilia A. And it was a presentation of the safety and factor VIII activity results. It was presented by Dr. John Sheehan from the Department of Medicine, University of Wisconsin-Madison. Now, the goal for gene therapy is to eliminate the need for regular factor VIII infusions through long-term endogenous expression of factor VIII. And so to do that, we have to deliver a good copy of the gene, generally targeting the liver, and then we're looking for expression that's going to be durable at a level that's going to allow patients to come off of prophylaxis. Now, as you've probably heard, there's lots of even late phase gene therapy trials going on for both hemophilia A and hemophilia B. So one question I've had about this program is, what's the need for yet another gene therapy program? So I think there's three core issues that this study is trying to address. So the first thing is, we know that between 30% and 70% of patients may have pre-existing neutralizing antibodies that are specific for different AAV serotypes, and this will preclude their eligibility to participate in gene therapy. So it is good to be exploring alternative vector serotypes and bring them into the clinic to see if this broadens the eligibility for patients. So that's the first point in this research program, is this unique HU37 vector capsid. The second thing is to get good expression at the lowest dose of vector possible. We really want to have strong targeting to the tissue where the gene's going to be expressing the protein. The liver's the target for hemophilia gene therapy. So this particular AAVhu37 serotype was chosen because it can specifically target the liver and hopefully do so with limiting the amount of vector that's required to get that good expression. The other key thing that was done in this study is they looked at a number of combinations of the enhancers and the promoters that are driving the expression of the factor VIII transgene. And they actually screened 42 combinations based on looking at liver specificity, the peak expression of human factor VIII that could be achieved, and then the duration of transgene expression. Now, of course, they did all of that selection of those different combinations in the preclinical animal models. So this is now first in man to see how that translates to the human experience. The third component was the immune response to the vector has been a real limitation of many of the gene therapy programs. And if we can't control that immune response with corticosteroids, it risks losing the expression because of the cytotoxic effect on the expressing cells. So the idea that for this more potent vector with high tropism and optimized expression is if we could keep the vector dosages lowest possible and still get good expression, this may limit some of the immune response. So what they presented in this study was a dose escalation study trying to find the optimal dose for the expression targeted for these patients. And the goal here again is to try to get patients so that they no longer need prophylaxis. They started at a relatively low dose of 0.5 times 10 to the 13th vector genomes per kilogram. And then they escalated to the second dose cohort, which was dosed at 1.0 times 10 to the 13th vector genomes per kilogram. And what was observed is even at the lowest dose cohort, both patients for a period of time were able to come off prophylaxis. The first patient got above 5% and was off prophylaxis for a number of weeks. And then eventually, because of some pre-existing joint issues, he had to go back on prophylaxis to ensure that he didn't have any ongoing joint issues. The second patient actually had a much more robust expression up into the 20 to 25% range. And actually, neither of these patients had any evidence of a liver immune response, and neither of them required use of corticosteroids. Interestingly, the second higher dose cohort, both patients have been able to come off prophylaxis, and it's been are essentially not requiring regular factor use anymore, and demonstrating a good bleed control. And one of the patients in the second dose cohort was able to get expression that is actually above the hemophilic range, in the 40% range. Now, this patient did have a very transient rise in the transaminase levels, which might have been an indication of an immune response. He was promptly put on a course of prednisone, and that completely resolved the transaminitis. And he actually has had sustained expression of factor VIII at a very good range, somewhere between 25 and 40%. So I think what this program is showing that is that it's hopefully achieving the goals here of being able to use a relatively low vector dose, do so obtaining good expression, allowing patients to come off prophylaxis, good bleed control, and hopefully a minimum of patients who are going to require any sort of immune suppression. So I believe that they're going on to the next higher dose cohorts to see if they can optimize the greatest number of patients being able to have a good response to this technology. So I do think it's good that we're continuing to explore these new alternative capsids, looking for other ways to optimize these technologies. And I think it's going to bring more choices to the clinic and hopefully broader eligibility for our patient population.